On the cover of Science Times this week, a startling look inside the brain. Edvard and May-Britt Moser are neuroscientists whose discoveries are changing the way we think about how we remember where we've been. They and their work are profiled in Science Times by reporter James Gorman, and they join us now from their laboratory in Trondheim, Norway. Hello.
Hi.
Thank you very much for joining us. Can you talk about a little bit about your research and what you have learned?
Yeah, I don't know. I could start and maybe Edvard could follow up, but at least it's quite exciting to understand how behavior is created by the brain. And then since we wanted to study rodents or rats, then to study spatial navigation is very easy. And then we knew about the work by John O'Keefe in London, and we also collaborated, or we even went to him, and he was supervising us in how to study spatial navigation and studying single cells in the brain and how they were related to spatial navigation.
Yeah, so what John O'Keefe discovered already in 1971 was that there is a type of cell in the brain that is active only when you are in certain places in the room. And those cells were called place cells. And what we wanted to find out, and which we have worked on when we started our own lab, is how do these cells relate to other cells in the brain? Are there other types of cells that also code for space? And how do they all work together? And we're trying to use that as a way to understand how the brain or the cortex of the brain is working in general, trying to find general principles. And the advantage we have is that these types of cells, as May-Britt says, are really easy to measure. So you can actually use them as a window into the deep parts of the brain.
Talk a little bit about the animals in which you do your research.
Yeah, so we're using mostly rats, also a little bit mice. And what we are able to do is to use very thin sensors that are implanted into the brain so that we can actually almost navigate these sensors between the cells and then listen to their natural activity, pick that up and then amplify it some 10,000 times. And then we can actually see on an oscilloscope the activity of individual neurons when the animal is finding its way through the environment.
And not only see, but we can even hear it. And the sound is like music in my ears because it's like popcorn. So whenever the rat is outside the place field in an environment, then the cell is silent. But when it starts to go into a field, then you hear pop, 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 pop. And then you go into the field and then the pop, 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 pop. Like you put a bag with popcorn in the microwave oven and you start to hear now it's pop, pop, pop. And then it's so frequent that you have to take the popcorn bag out. Then you know you are in the middle of the place field in the environment.
One of your key discoveries involves something called the grid cell. And I'd like to ask you to talk about what is a grid cell and what does it have to do with our memory of where we are in space?
So grid cells are cells that are active only in certain places in the room where you are, but many places. And the fascinating thing is that those different places where a cell is active form a very regular hexagonal pattern, so that it consists of almost like triangles, equilateral triangles that repeat over and over again. So it's almost like a matrix or like a graphics paper that you put over the room, and then the cell fires only at the cross sections. But the difference from graphics paper is that it consists of triangles that then repeat over and over again. And this kind of map, it's almost like intersecting lines. It contains very accurate information about where you are in the environment in meters and angles, so distances and directions. And we know that this information is there, and it's very likely that the brain actually then uses it also to measure where you are in space.
Where do you see your research leading? Will it help in diagnosing and treating disorders of memory like Alzheimer's?
Yeah, what is exciting is that when we found these grid cells, we found them in a place in the brain that is called the entorhinal cortex, which is connected highly to another place in the brain that is called the hippocampus. And if you lose the hippocampus, you can't get new types of memories about events and facts. And if you have a tiny damage in the entorhinal cortex, you can't find your way. And you can't find your way if you have a damage in the hippocampus also. What they have shown is that there are some groups of Alzheimer's patients that have a tiny lesion in the entorhinal cortex, exactly where we found the grid cells. And if they start to see this lesion, they can say whether this person is going to develop Alzheimer's disease or not. And the first symptom these patients typically have is that they can't find their way in the environment. So the spatial navigation system is out of function.
So very important work you're doing. Edvard and May-Britt Moser lead the Kavli Institute for Systems Neuroscience in Trondheim, Norway. I want to thank you both for coming on the podcast and telling us about your research.
Thank you.
Trondheim, Norway. In 1988, two determined psychology students sat in the office of an internationally renowned neuroscientist in Oslo and explained to him why they had to study with him. Unfortunately, the researcher, Per Oskar Andersen, was hesitant, May-Britt Moser said as she and her husband, Edvard I. Moser, now themselves internationally recognized neuroscientists, recalled the conversation recently. He was researching physiology and they were interested in the intersection of behavior and physiology. But, she said, they wouldn't take no for an answer. We sat there for hours. He really couldn't get us out of his office, Dr. May-Britt Moser said. Both of us come from non-academic families and non-academic places, Edvard said. The places where we grew up, there was no one with any university education, no one to ask. There was no recipe on how to do these things, and how to act politely, May-Britt interjected. It was just a way to get to the point where we wanted to be. But see now, when I know the way people normally do it, he said, smiling at the memory of his younger self, I'm quite impressed. So, apparently, was Dr. Andersen. In the end, he yielded to the Moser's combination of furious curiosity and unwavering determination and took them on as graduate students. They have impressed more than a few people since. In 2005, they and their colleagues reported the discovery of cells in rats' brains that function as a kind of built-in navigation system that is at the very heart of how animals know where they are going, and where they have been. They called them grid cells. I admire their work tremendously, said Eric Kandel, the Nobel laureate neuroscientist who heads the Kavli Institute for Brain Science at Columbia and who has followed the Mosers' careers since they were graduate students. John O'Keefe of University College London, whose discovery in the 1970s of so-called place cells in the brain that register specific places, like the corner dilly or grandma's house, and who was one of the Mosers' mentors, said that the discovery of the grid cells was incredibly significant. The workings of the grid cells show that in the brain, you are constantly creating a map of the outside world, said Corey Bargmann, of Rockefeller University, who is one of the two leaders of a committee set up to plan the National Institutes of Health's contribution to President Obama's recently announced neuroscience initiative. Often, the workings of billions of neurons that produce our thoughts are opaque. But electrical recordings of signals emitted by grid cells show a map, with the framework and coordinates that are completely intuitive, Dr. Bargmann said. And to find such a straightforward system is, in its own way, just mind-boggling. What is the brain doing being so mysteriously unmysterious? The implications of the discovery are both practical and profound. The cells have been proved to exist in primates, and scientists think they will be found in all mammals, including humans. The area in the brain that contains the grid cell navigation system is often damaged early in Alzheimer's disease, and one of the frequent early symptoms of Alzheimer's patients is that they get lost. The Mosers do not work on humans, but any clues to understanding how memory and cognitive ability are lost are important. On the most profound level, Dr. O'Keefe, the Mosers and others speculate that the way the brain records and remembers movement in space may be the basis of all memory. This idea resonates with the memory palaces of the Renaissance, imagined buildings that used spatial cues as memory aids. The technique dates to the ancient Greeks. In this regard, neuroscience may be catching up with intuition. A welcome ambush. Edvard, 51, and May-Britt Moser, 50, now direct the Kavli Institute for Systems Neuroscience and the Center for the Biology of Memory at the Norwegian University of Science and Technology here in Trondheim. They have a steady stream of findings coming from their lab, and a slew of awards, the latest of which, the Pearl UNC Neuroscience Prize, they received April 16 at the University of North Carolina. But they did not grow up in a center of academic ferment or intellectual competition. They were born and raised on islands off the coast of Norway, a couple of 100 miles north of Bergen, part of an area known as Norway's Bible Belt. They went to the same high school, but didn't really get to know each other until they met again at the University of Oslo in the 1980s. May-Britt, who grew up on a farm, remembers an environment in which drinking, card playing and dancing were all frowned upon. When she called home from Oslo announcing that she had been to a bar and had her first beer, her mother said, "And what's next?" The Mosers married in 1985 while still undergraduates. By the time they had finished their doctorates, in 1995, they had 2 daughters, but they were ready to see the world, to train in laboratories outside Norway. And they did spend time in England, with Dr. O'Keefe, and in Scotland, with Richard Morris at the University of Edinburgh. But the Mosers' travels were cut short when they were ambushed by a job offer too good to refuse, from the university in Trondheim, where they have been ever since. Without knowing it, we actually negotiated, May-Britt said, because we were not interested if we only got 1 job, and we got 2 jobs. And we were not interested if we did not get the equipment we needed, and they gave us that. Suddenly, without having really planned it, they had their own lab. Of course, nothing happens suddenly in research. They began in what Dr. May-Britt Moser described as a bomb shelter, and gradually, over time, built up their program. Similarly, they did not set out looking in the part of the brain where they ended up. They began recording the activity of cells in the hippocampus, with electrodes implanted in the brains of rats as they roam an enclosed area. This is still a main method, and the rats are intriguing to watch, pursuing little bits of chocolate cereal on the floor of an enclosure, seemingly oblivious to the implants attached to their skulls. A black box. The Mosers wanted to find how information was flowing to the play cells, whether it was going from one area of the hippocampus to another. But even after they inactivated sections of this brain area, the play cells still functioned. So it seemed that information was flowing in from the nearby brain area, the entorhinal cortex. They started looking there, and in their early work they were helped by Menno Witter, then in Amsterdam, now at Trondheim, in the delicate task of guiding the electrodes to the right spot. We didn't immediately find the grid cells, Dr. Edvard Moser said. At first they noticed cells that would emit a signal every time a rat went to a particular spot, and they thought that perhaps this was something like the play cells in the hippocampus that are tied to locations in the outside world. But gradually they learned that what they were seeing was a cell that tracked the rat's movement in the same way, no matter where the rat was. The cell was not responding to some external mark, it was keeping track of how the rat moved. And when they gave the rats enough room, a very regular pattern emerged. The first thing was that we thought there was something wrong with the equipment, Dr. Edvard Moser said. I thought, is this a bug? Dr. May-Britt Moser said, after a 2005 paper in Nature, in which they reported the discovery and named the cells. Other labs confirmed the findings and more discoveries followed, in their lab and elsewhere. It is now clear that the grid cells, in combination with cells that sense head direction and others that sense borders or boundaries, both originally identified in other parts of the brain by other labs, form a kind of dead reckoning navigation system in the brain that maps movement. Information flows from this part of the brain to the hippocampus, and then back. Exactly how the grid informs the place cells, and vice versa, is not known. What scientists have now are two ends of a system with a black box in the middle that is not fully understood. At one end are place cells. At the other are grid cells. As to what exactly happens in between, and how the grid cells form in the first place, Dr. Edvard Moser said, that's still a 20-year research problem. Or, as Dr. O'Keefe put it, we are still in the pre-Newtonian phase of neuroscience. The Mosers remain something of an anomaly. Not only are they off the beaten academic track, but they are a married couple who work together on the same scientific problems at the same institution at the highest levels of science, a true rarity. They do have different spheres in their new, state-of-the-art lab. May-Britt is more hands-on with the experiments and the design, and Edvard is more involved in mathematical analysis and interpretation of the results. We have a common project and a common goal, he wrote in response to an emailed question, and we both intensely yearn for it. And we depend on each other for succeeding. He continued, most couples manage to cooperate on child raising. For us, our brain project is our third child, so nothing different, really.